Okay, welcome back after lunch. Hope we've all fed and watered and ready for another exciting session. So thanks to the organisers and Miltenyi for inviting me here to share some of my experience with you briefly this afternoon. The way that we're going to play this, I've got a few slides to go through as a bit of a scene setter, and then have a conversation with Dmitry from Miltenyi about how the field is developing. I'm Chief Development and Manufacturing Officer at adthera Bio. My background is a little over 20 years in manufacturing in the cell therapy space, historically in an academic centre with NHS, but servicing everything from first-in-man academic programs through to commercial supply. We set adthera up a little over 2 years ago because I recognised there's a gap between those academic centres and the very big CDMOs on the planet, and we're trying to help those early biotechs get things right first time. As we know, most of what has come into the cell and gene therapy space is developed out of academia, and that will continue to be the case for the foreseeable future. Even the pharma industry now that's buying into the space is acquiring biotechs that have come out of academic centres. And that's different from other aspects of pharmaceutical development. The other difference we see is that in the analytics space, it's academic research that breeds these processes that we use, these techniques we use, these modalities for assaying, for evaluating, and the next generation of assay platforms is going to come out of the R&D space of academia. So we have to understand how to capitalize on those developments while still making sure that they maintain the regulatory and compliance needs of our industry. And where I usually begin a journey with a client is with a process that starts looking something like this, and very open and very manual in a biological safety cabinet in a GMP suite, but very little closing of process and very little thought of how the analytics fit together. What we tend to find is people want to measure everything. And that, I understand, we want to understand our product. But there's a balance between measuring for value and measuring for comfort. And there's a lot of stuff that we find early days, there's redundancy in measurements that is a comfort blanket for the developers rather than actually bringing value to the product. So our job is to close those processes. Now, that can be a modular solution, that can be an all-in-one platform. But if we close those processes, how do we know what's happening inside our bioreactors? And more often than not, what we assay is what comes in and what comes out. And in the middle, we may take samples, there may be some key gateways that we have to go through, but largely, we know what we start with, we know what we end with. If they meet specification, then that drug product can go out to the patient. So how do we build better processes and systems to allow us to understand more completely what's happening in our bioreactor platforms? Well, we can use smarter platforms. Miltenyi Biotec Clinimax Prodigy gives a solution essentially for end-to-end. We can now go from start material to fill and formulation on a single device. Within its body itself, it doesn't have analytics capability, but with software updates allowing us to understand what the machine is doing and be able to control the machine remotely, we can segue samples that come off that device into the QC lab without having to have operators there manually taking material from the device. Others in the space, Cytiva, have their Cephia family for wash and labeling, now selection and expansion. Again, a modular solution, but potentially an end-to-end package. The integration with external systems is really good with Cytiva, but it's a new piece of kit on the market, so we're still learning how best it can be utilized. Others have brought more analytics into the platform. The Cocoon has some. adthera probably is the most advanced at having metabolic measurement devices built into it, but it can't do the selection piece, so it's a trade-off. We can do the bioreactor, we can understand what's happening inside, but we can't go from end to end as we can with other devices. Similarly with Lonza, we're not going to start with a Luca pack. And then there's next-gen devices now coming to the market. So Ori have launched their Eero, again, a bioreactor solution, but very well digitally connected and designed in a way that the operator can be minimally skilled compared to perhaps some of the other platforms, and interact with it in a very simple manner. Importantly, can be controlled and monitored and programmed remotely. Or then there's solutions like 908 devices, so the guys that produce the Maven platform, where you can introduce those probes into your bioreactor, into a system in some way. So if the system itself doesn't have the means for measurement, can we introduce a means for those process analytical technologies to be integrated into our platform from a third party? So we can probably figure out ways to get samples in and out or measure on the device. But where do we go next? Where do we go next in the automation process? Well, we've got companies like Cellular Origins that are taking those modular solutions and providing a robotic solution to manage them as a human would. Great to be able to accelerate the process in the field, because we're not having to invest in a de novo novel device. We can still work in a G-Rex with a Lovo and have the Cellular Origins robot take over the human aspect of it. That allows us to bring more scale to our manufacturing, but also allows us to automate those samples that are coming out that can then move into a different space in the manufacturing suite to provide that continual monitoring. And then we go to the far extreme, where we've got companies like Solaris who are reporting to have a device that is completely end-to-end. And I say that with a little bit of a we wait and see. It's clearly developed a lot. There's good investment in it. We've now seen more of the platform and how the consumable works, but this is a device where we're now having to move our process entirely onto a novel platform. So we're not going to be able to say, okay, I've done my phase 1 and 2 on a G-Rex, and now I'm going to automate it with a robot. I'm moving to an entirely different platform, so I'm going to have to go back and do some of those early studies. So there are countless ways that cellular material and data flows in and out of our processes. But what about the analytics that we don't want to do on the hardware itself? So the vast majority of what we do in our space is flow cytometry based, certainly for identity and phenotype. We've got Acelix here today, really great platform, simple to use, single hit cartridge, sample goes in, machine does the analysis, data comes out. That can pump into a digital system, entirely hands-off by the operators. And then Miltenyi's solution with the MaxQuant gives me the flexibility I want today. I can do all my development work, I can sit there at the machine and validate assays, and then we can work with Miltenyi to build a custom solution so that assay essentially works in the same way as the Acelix platform. So the MaxQuant will do the analysis, we'll do the gating, we'll do the data export and push it straight out to my digital solution. So the operators aren't having to manually gate and decide where they should be putting their analysis windows. And then there are other aspects of the manufacturing process, things like mycoplasma, so Biomeria's solution for rapid mycoplasma, or the Bactalert system for sterility, through to quantification of antivirus and vector manufacturing, things like Unchained Labs, Leprechaun. They've got the greatest suite of named equipment on the planet. The point is, all of these things provide data points, and this is a token gesture of a few. There are 20 or 30 different platforms out there that we want to be able to acquire data from. So we've got all this data. How do we then use digital solutions to unblock the bottlenecks that we know exist? And those sources of data are vast, and this list is not an exhaustive list. I've highlighted the two around analytics and manufacturing because that's what we're here to talk about today. And this is where at adthera we're taking an approach to bring digital solutions to allow us to use the data in a way that's more efficient. And what I mean then is that digital systems should not be a paper on glass solution. So just because I can take a piece of paper and make it appear on an iPad, that's not digitization. What we need to be able to do is take that data and use it in a way that's smart. And what we're building is a cloud-based solution that means that data can flow from different manufacturing sites through an adthera integration layer into our central control hub. So we can see what's happening at each manufacturing site, at each bioreactor, at each patient process in the manufacturing space. But that also then segues into the chain of custody, chain of identity, logistics partners. So the end-to-end supply chain becomes an entirely digitized solution. And we believe by doing that, we can bring the scale to the market that we need without having to centralize everything. We can keep to a decentralized solution and have data flowing in and out of a hub. And the way we see it looking, it's slightly blurred out because we haven't patented the name of it yet, so I can't disclose it. But there's a front end that it could be a manufacturing partner, it could be a clinical partner, it could be a patient. We want to give people access to the information that they need. Now, clearly, patients don't need to know the MOI of a vector that we're using, but they might like to see, hey, where's my process at? You know, you order a parcel today from Amazon, you get a little tracking beacon that says your DHL man's 54 drops away. You go and have a therapy that is transformative for your life, and you have a pretty invasive apheresis and walk away for 6 weeks and don't know what's happening. You know, how do we give something back to the patient so they can see? So the portal will be utilized by different clients, and it can give you everything we need. So that could be a CDMO partner. They get a snapshot instantly of what's happening at all the manufacturing solutions. Where are the batches at? You know, can we see if there's a hold, if there's a delay? Trending, how do we see how batches are varying over time? So the data that we've got coming through the system is there to be able to give evidence to the quality teams and the regulators that the product meets spec, but using it smartly allows us to do a lot more than just collate data. So I'm going to stop talking now there and invite Dmitry to the stage, and we'll have a bit of a conversation about how we see this space developing in the future.
Yeah, thank you very much, Stuart, for this lovely talk and for sharing with us your integration vision. And it's a good point that you actually mentioned that we need to unblock these digital bottlenecks, basically to make this information that is crucial to you, crucial to your partners and patients in the end, to make it available, right? And what we want to do for that, as in the case of Miltenyi at least, is to create for you a gateway, a single gateway where you can actually retrieve information from all of our instruments. So we are not going for single instrument integration as we planned it, but we are going for a gateway that basically knows how to talk to the external systems by integrating to that gateway different protocols. Like you might have one protocol preference, another customer has another protocol preference. So that's what we want to basically create, right? Like a gateway that will allow you to integrate our systems all at once, the ones you have, independent whether it's Clinimax Prodigy or whether this is MaxQuant, right? So once you have this gateway, you can basically get the information that you basically need, because to unravel this buzzword, digital integration, what does it mean actually? It basically means in the end that you want to have probably a dashboard, for example, where you can see alarms and notifications coming from the different single instruments. We heard previously from our colleagues here that we are talking about 40 plus batches a week, for example, right? This is not a setting where you really want to go to the instrument, into the facility and look at a single instrument, what is going on. You want to have an actual overview, basically. So, yeah.
And I think the gateway is a great solution. And I think what's really important is you allow clients like adthera to use the gateway to feed our display system, rather than have to use a dashboard from yourselves. What we don't want is to have a Miltenyi dashboard, a Lonza dashboard, a adthera dashboard, a Ori dashboard. That,
yeah.
You know, yeah. If a client's only using that device, great. But having the means for that portal to be able to export its data to a different display tool,
right,
that will be our proprietary, will be.
Yeah, so you basically what you want to have is your own dashboard, for example, where you want to send a command from to one of the devices, the gateway translates it, and you get the information back that you actually need.
Yes. Yeah. But we don't have to invest the effort in integrating every individual device in the Miltenyi portfolio to our dashboard. Yeah. We'll just integrate your
one piece, one single
back
piece.
To our
hub. Yeah. No, that makes sense.
But actually the limitations today are the time it takes humans to interact with the devices and feed them and take the data and move the samples, and then that we see a 5-day process taking 3 weeks for quality to be able to review paper records. And in my past life we had 1,000 pages batch records that had to be reviewed, and there's going to be deviations because humans can't write when it's 3 o'clock in the morning and they've been in a clean room for 12 hours and they want to get out and things have gone wrong. Computers don't make those mistakes. So once we've validated that platform, the usability becomes less of an issue. We can run simultaneously 10 prodigies, 20 prodigies, 50 prodigies, 1,000 prodigies. There's no reason we can't be having a distributed manufacturing network across the whole of Europe, simultaneously manufacturing 300-400 batches of product.
Yeah, and actually another topic in regards of manufacturing and QC, since we are talking about both, as I understand, is the wish of the industry also to not have to transfer your sample while you're manufacturing in process, right? You probably are looking for, let's say, one device doing so and inline QC. Is that something you also envision for adthera at some point?
You mean in process analytics?
Right.
Yeah, of course. I mean, if we could take away the need for the QC lab to have to do each of those assessments, then yeah, that will bring savings clearly. I think the more we grow as an industry, the more we understand our product, we'll be able to decide where we can rationalize what we do measure and what we don't measure. I still see today clients that come with assays that have been there from day one because they had them, because they at the time thought maybe we should. Mycoplasma is a good example. We see mycoplasma assayed a lot in a 2-day autologous program. What am I going to do to the mycoplasma that probably didn't exist to start with, and if it did, it was in the patient already in 2 days? I'm going to do nothing to change it, but it's still there. It can be a slow assay. If it's the old-fashioned culture method, you're taking 30 days to get data back to be able to release. So if we can do smarter things in process, that means we can consolidate what we have to do offline at the end. We're always going to have to assay drug product. That's not going to go away. But if we can do more as we move through the process, so we streamline that release piece, we can get drug back to patients quicker.
And another point on the QC part, since NGS is evolving a lot in the QC, and from our early conversation I understood that probably NGS is very interesting to look at because you can detect whether you have a CAR-positive cell most likely in an earlier stage, right? Because you don't have to wait for the expression and the flow, but as I understood you previously, you said, but this is still not giving us the functional assay, right?
So again, right now we don't know enough about how those individual CAR T cells are behaving. Are they killing a single target, engaging, and then not disengaging, so they kill with a one shot, or are they serial killers? And that makes a difference to your potency. Now NGS isn't going to tell you that. That's a biological assay that needs to look at how the cells behave with a target. So are we never going to see NGS replace it? No, I don't think that's true. I think at some point we'll understand enough of what we can measure as a surrogate that tells us, okay, we now don't need to do those physical assays, but I think we're 10 or 15 years away from a solution that doesn't have a cell trying to kill something in a well.
Yeah, thank you very much for the lovely discussion, and we probably give the chance to the audience to ask some questions as well.
So we can electroporate on the Prodigy, whether it's the best in class, we'll leave for another day. We don't see a single platform solution, to be clear. It was just an example that the Prodigy in principle can go end to end. What we see is a bit the same as our approach to our digitization, picking the best in class for the job it needs to do. And that's where the portals are really important. That all we need the manufacturers of kit to do is to give us the tool to be able to see what's happening and communicate. I don't need every company to give me a batch record solution. Just give me the means for the data to come into my digital batch record. So we can do that with, you could be on a Prodigy, we can be pulling data out of Fresenius Q, we can be pulling data out of, who else have we played with? Lonza, we can pull data out of adthera. So it doesn't really matter as long as there's a pipe to get the data from the machine to the digital hub and vice versa, ideally. So we can tell the machine what to do after it's told us where it's at. The rest is, I say, our job. I haven't got a clue how we do it. We've got digital brains that understand this. But the computer whizzies can build systems that allow that control to happen. And when it all starts to work well, the savings are incredible because you don't get the errors that you see in paper batch records because the system's validated, it can't make a mistake. But you also see things as they happen real time. Whereas, let's say we're running an assay that's 4 or 5 days, and or a manufacturing process 4 or 5 days long. Operators will come in the morning and, oh look, the system's an alarm, or when did the alarm trigger? It went off at midnight last night. What's been happening for 8 hours? I have no idea. The digital systems instantly tell you, device is telling me X. Okay, quality team look at it, proceed. And the system carries on, because 9 times out of 10, those errors are yellow errors. Something's happened I wasn't expecting. Okay, are you sure? Try again. Oh, I'm happy now, carry on. Rarely do we get errors that are catastrophic in the manufacturing platforms. They'll still happen, and then you'll still have to go in and rescue. But I think having the portal being able to talk back to the manufacturing platforms, then you can be truly modular. The challenge then is showing consistency at all of our sites. And from the analytics bit, that's a bit more of a challenge, but that's why things like Acellix or Miltenyi's Stain Express with smart gains and using the customized protocols, we can show that 2 mat squants at different locations are identical and gives exactly the same data on the same cells at the same time. So we can take away that variability in analytics between sites. So we're building a single integration layer. So as long as we can make everybody talk to our integration layer, what we do with that downstream is a little bit like Miltenyi's portal. They can all talk however they like. As long as what comes out of it is a language I understand, I don't need to care how the bits are happening upstream. And that's important because things will change. If I directly integrate without a layer, I've got to validate that pathway. If that system changes, I've got to validate all the way back down to my portal. If I have an integration layer, everything north of the integration layer needs to be functional, but as long as the language that's coming out of it is consistent, I don't have to revalidate that each time. Now, all the companies we're talking to are already have a solution for us, or at least a long way down the road of delivering it. It varies. So some will be, here's an API and that's the pipe. Others, Cytiva are a good example. Cytiva go everywhere from, here's our solution, here's an API, here's my code. Go and write your own system. That's probably a little bit too extreme for us, and we don't want to be rewriting what already exists, but if the system has a human-machine interface, you can make it talk, because it's talking to the screen. So all you've got to do is make that message go to a different location, and that's entirely feasible with everything on the system, everything on the planet today we use that has any kind of digital interface can talk to one of these portals. It's just getting the companies to open the pathways, and we're getting more and more that are doing it. So I think those that don't will fall by the wayside. Okay, thank you.